What you're going to be doing for the next couple of classes up until the next little exam is you're going to be practicing working with phylogenies, reading them, getting information out of them, but ultimately what we're going to be doing is reconstructing phylogenies, taking the characteristics, the clues that we find in modern day organisms, and using those to draw inferences about the evolutionary history of those organisms, who is most closely related to who, and also to draw inferences about what the ancestors of those organisms were like. And biologists have managed to do some pretty amazing things with these tools. It's almost like having a time machine and being able to go back in time and look at organisms that have been dead for millions of years. So what is the goal when we're doing this? What is the goal when we're trying to reconstruct a phylogeny? There's a term that was introduced in that little video that you watched, the one that I didn't make, at the very beginning of this module, and it's a clade. That's going to be an important concept, so I want to kind of reinforce what a clade is. Here's a phylogeny um, that shows various different kinds of groups. A clade is a monophyletic group, which is to say it is an ancestor and all of its descendants. Okay, any ancestor and all of its descendants are a clade. So for example, what's shown in this blue box is a clade. There's this ancestor here and all of its present-day descendants. Okay, if you took this ancestor here and you put a big box around it and I and J, that would be another clade because it would contain an ancestor and all of its descendants. If you started here and put a box around G and H and I and J, that would be a clade because it could contain an ancestor and all of its descendants. Okay, so that's one thing that's always true about clades. You can nest them within one another. Okay, another thing that is true about clades, and this is just useful for knowing whether or not you are looking at things that you're looking at as a clade or not, is that if you've got a group that is a true clade, you can isolate it from the rest of the phylogeny with a single cut. In this case, the cut would be right here, and if you cut that off, you'd be holding in your hand this branch, which contained H and I and J. And it should make sense to you intuitively that a clade, an ancestor and all of its descendants, right? A clade is something you can always remove with a single cut because a clade is basically just an ancestor and everything downstream of it. So when you cut right behind that ancestor, you're getting the ancestor and everything downstream of it. There are a couple of ways to make groups that are not clades. Here's one. You take an ancestor and some of its descendants. The only way to get B and C and D in a group together without A, though, is to cut here and here. Okay, so you can't do that with a single cut. That means that it's not a clade. It's not an ancestor and all of its descendants. Another way of doing getting a group that's not a clade is to include a group of present-day species but not include their common ancestor. And technically, these two different groups have different names. This one's called paraphyletic, that one's called polyphyletic. I will never ask you about these names. I don't think they're important, because what really matters is it a monophyletic group or not? In other words, have you got a clade? Another thing that's true about clades, I mentioned that they can nest within one another, but two clades cannot overlap. Okay, we'll see why this is important later on. When you are looking at the evolution of traits or the evolution of organisms on a phylogeny, what you really have to do is focus on specific things that are changing. And particularly when you are reconstructing a phylogeny based on the information that you can get from present-day organisms, what you have to do is you have to first define what characters you are looking at. A character is any feature of a group of organisms that is thought to be derived from or evolved from a corresponding feature of their common ancestor. Okay, and usually what you want is a feature that is found on all of the organisms in your in-group. That's what's going to be useful. A character state is one of any of the possible conditions of this character. Now this is super vague and abstract, so let's put some concrete examples to it. One character might be integument, the covering of the outside of an animal's body. Different character states that you might see could involve scales, they could be fur, or they could be feathers. So a character is a type of thing, and the character states are specific instances. It's like, the character is ice cream, character state is different flavors of ice cream, except in this case we're talking about scale and fur and feathers, and that doesn't taste nearly as good. More often than seeing these morphological characters, these days what you're likely to see are phylogenies that are built on molecular characters, in other words, genetic sequences. In this case, you'd be looking at a particular locus, and a character would be whatever nucleotide is at position 1612 at that particular genetic sequence. It could be an A or a C or a G or a T. Those are the possible character states. And so each position in a particular gene, in fact, every nucleotide in an entire genome is a separate character. And presumably, obviously I'm not going to go into great detail about how this works. There's a lot of complexity in this, but presumably this nucleotide here in the mouse and this nucleotide here in the fly were at some incredibly distant time in the past copied from the very same piece of DNA. So these nucleotides really are, in the most literal way, derived from a corresponding feature in a long, long ago common ancestor. Okay, I've been nattering on about clades. Why are clades important? Why should we care about them? Clades are central to the logic of how we reconstruct the evolutionary history of a group of organisms. When you're looking at a whole bunch of organisms, you want to know how they're related to one another. The way you figure that out is by thinking about clades. And here's why. You've got a lineage that is evolving along. It's got a particular character, and at some point in its evolution, it evolves a novel character state. Okay, it may be that that lineage is then going to evolve on, and it's going to give rise to a bunch of different species. But because that ancestor back here had that novel character state, the simplest outcome, the most likely outcome, is that all of its descendants, all those species that it gives rise to, are going to inherit that new character state. So for example, here's a phylogeny of the vertebrates. What's shown here is the evolution of keratinous scales. Keratin is the protein that makes up your fingernails and your hair, but the reptiles have scales on their epidermis that is made of keratin. That includes lizards, it includes some things that aren't shown here like snakes and tortoises and turtles. It also includes the crocodilians and their close relatives, the birds. If you didn't know that birds were really most closely related to crocodiles, well, that's a newsflash for you. The reason that all of these organisms share this particular character state is because they are all descended from this long ago common ancestor that was the first lineage to evolve that particular character state. And it went past that character on to all of its descendants. The reasons that organisms like fish and salamanders and mammals do not have scales made of keratin is because they are not descended from this lineage right here. Fish have scales, but they're actually bony scales. They are formed from the dermis rather than the epidermis, so they're actually quite different from the scales that you see in reptiles and birds. This logic that most often a lineage that evolves a novel trait or a novel character state will pass it on to all of its descendants is the foundation of the logic that we use to reconstruct organisms' evolutionary histories. So the problem that you get into is that when you look at a group of present-day organisms, you see different character states. You see, for example, some organisms have scales, some have fur, some have feathers. And what's important when you look at these character states, what's important is that these characters all be homologous. They all need to be derived from the same structure in a common ancestor. And here's an example of homologous structures in vertebrates. This is the vertebrate forelimb. Remarkable diversity of form among vertebrates in the way that their limbs are put together. Here's the human forelimb, here is the front leg of a dog, the front leg of a horse, and the front flipper of a whale. The bone structure has been radically changed, but this structure is derived from the same structure in the common ancestor of all of these mammals. So the earliest mammals had forelimbs that started with one bone, that then split into two, then you get a lot of wrist bones, and then 5 of these digits up here. And all of these other species have the same basic structure. In the horse, most of the digits have been lost. In the whale, one of the digits is so reduced that it's barely visible, but the genetic architecture underlying all of this is all the same. In contrast to this, you can have characteristics that look similar, but evolved from very different structures in a common ancestor, or maybe just evolved completely independently. They're not derived from a homologous structure in the common ancestor at all. So for example, moths have wings, pterosaurs have wings, birds have wings, and bats have wings. But all these different kinds of wings evolved independently. So they don't share what's called homology. They are analogous. One of the things you have to be very careful of when you're reconstructing a phylogeny is you have to be sure that the character states that you're looking at, the structures you're looking at, are actually homologous. If you try to build a phylogeny based on analogies, on analogous traits, it's going to go very badly wrong and it's going to be very confusing. We'll see why, I believe, in the next set of videos, not the ones that you're going to watch today, but the ones you're going to watch in a couple of days. So one of the riddles that scientists have to solve is they have to figure out which characters are homologous. And this might seem pretty daunting. When you look at all those different vertebrate forelimbs, they don't necessarily look all the same. How do you know that they're actually derived from the same structure in the corresponding structure in a common ancestor? Well, often, even if a structure looks very different superficially, if it's homologous across multiple species, there will be these developmental clues which tell us that, oh, these evolved from the same ancestral structure. They'll share developmental pathways in common. So for example, when I talked about the vertebrate forelimb, notice that there's one upper arm bone, 2 lower arm bones, a set of carpals, and then 5 digits. And that's going to be true about no matter which vertebrate you're looking at. That underlying similarity suggests that that's a homologous character across all of the mammals. Similarly, when you look at, say, different birds, you can see clearly you've got the same bone structure in the wings of each one. And that suggests that wings are a character state that across all these different birds is homologous. When you look at the structure of bones in a bat wing, however, it's clear that this wing has a very different structure. It seems to have evolved separately. There are these very long fingers instead of the short fused ones. The surface of the wing that catches the air in a bat is this stretched out web of skin, whereas in birds, the air-catching surface is feathers. There are all sorts of developmental differences here that you don't see among the birds. At the level of a forelimb, these are all homologous because they all share things like the same bone structure and a certain genetic architecture underlying it. But as a wing, these evolved separately. And you can see all of that in the developmental clues, the things they have in common and the ways they differ. In some cases, you can tell which traits are homologous in a genetic sequence by aligning the sequences. So for example, if you have this lowest sequence here, and you're trying to decide which of these bases, this is actually an RNA sequence, which of these bases is homologous with which in these sequences, you can feed them into a computer program and it'll basically try to line them up in the way that makes the most sense. And once you do that, what you get is a strong sense that some of these characters, like this base here at position number 33, these are all homologous. Basically, there's a C here because there was some kind of mutation that changed it from a U to a C. The others have remained unchanged since they had a common ancestor. One thing I want to point out, though, is no matter how hard you try to establish which characters are homologous and which are not, inevitably you're sometimes going to make mistakes. Sometimes you're going to try to build a phylogeny based on characteristics that are not homologous. That does cause problems, but we'll see how to solve those as we go along. What I'm going to be doing for the rest of this lecture and the next is we're going to be coming back to the same evolutionary history. I'm going to show you what happened during the evolutionary history of a group of organisms. And then we're going to take just those present-day organisms, and we're going to show how, using the characteristics that are in those present-day organisms, you can reconstruct that exact same evolutionary history using some fairly simple logic. So a long time ago, there was a lineage that was evolving through time. And it split into two. There was a speciation event so that you now have two different lineages. One of them lost a feature that we call fins. Evolution continued. The lineage that lost the fins split again, and one of those daughter lineages lost these structures called gills. So originally you had an organism that had both fins and gills. This one has kept the fins and gills. This one lost the fins. We kept the gills over here, and now over here we've lost both fins and gills. This lineage also developed these things called nails or claws at the ends of its toes and split again. Of the daughter lineages here, one of them evolved scales that are made of keratin. This might sound familiar to you. The other one evolved a whole suite of different traits. It evolved fur, it evolved glands that give off this, this modified sweat, actually, that we know as milk. And it became warm-blooded. It kept its body temperature well above ambient. The critters over here with keratinous scales split into two groups, two lineages, one of which evolved this structure called a gizzard for grinding its food. Up here, we haven't been paying much attention to this lineage that kept the gills when all the others down here lost theirs, but it underwent a significant shift in its life cycle where it goes through a dramatic change in its body shape halfway through its life. Everybody keeps evolving through time. The organism with gizzards split, so do these warm-blooded furry creatures down here. And one of the lineages that comes off of the gizzard-bearing organisms evolves to be warm-blooded as well. So it evolves at a temperature that's well above ambient. And at that point, we just sort of, these organisms go ahead and evolve on to the present day, and this is what they look like now. Of course, when you're trying to reconstruct a phylogeny, you don't get that whole retrospective about what these lineages have been doing for the past however many millions of years. What you get are these species. But you can also look at them and look at their character states. So if we put in a table, there are all these different character states that I mentioned as we were going through or all these different characters rather, that I mentioned as we were going through that evolutionary history. And for each of these characters, some of these organisms have those particular characters or they lack them. So for example, this character here is mammary glands, and there are two possible character states. There is has mammary glands, and there is does not have mammary glands. So some of the species here have them, some of them don't. The same thing is true for claws and nails. There's have claws and nails, does not have claws and nails, and so on. This is the data that we have to work with if we're going to reconstruct the evolutionary history of this group of organisms. What character states do they have for each of a group of characters that they share in common?